We are uploading now again in the YouTube channel, that's why. Okay then, we'll go ahead. Good morning to all of you.
Good morning, sir.
Okay, and today we are starting a fresh chapter. This is chapter number 7, Structural Organization in Animals. And in this chapter, as I have already posted the PDF form to you, it is only animal tissues. Others are deleted for your batch, like last year batch. So only animal tissues will be the syllabus for your batch. You are not going to study the morphology and anatomy of earthworm, morphology and anatomy of frog, or morphology and anatomy of cockroach, all this. Okay. Just 1 minute, somebody is waiting. I will open that. Okay. So let me unlock the room. Right, now anybody can join. Right. So basically, when we say animal tissues, we are concerned about our own tissues, including human beings. What are all the tissues that are present in our body? So it is always good that we know about ourselves, so that anything change that takes place in that part, you know why it is taking place, what to do to stop that change. Okay, that is the purpose. Knowing about ourselves is far better than knowing about others. That is the thing. Okay, so we know the strength, we know our weakness. Build upon the strength and convert your weakness into strength. That is the thing. So basically, as Naveen said just now, this unit consisting of 2 chapters, morphology of flowering plants plus animal tissues. Together it is for 8 marks for your batch. So you can consider it as 4 plus 4, or it can be 5 plus 3. So that means not many 1 mark, 2 mark questions will be coming from this chapter. And since your first term has got MCQ, so lot many MCQs you can expect from this. So with that background, we will go ahead. All of us in junior classes, we have defined what is a tissue, isn't it? Tissue is a group of similar cells having specific function and origin. Here 2 points are important. Origin should be same. Remember, all of us have originated from single cell called zygote. You know, we may be now feeling that we are all great people, 6 feet tall, 100 kilo plus weight, all that. But what is our root? Root is only single cell. Irrespective of who we are, what we are today, our life started with single cell, that is zygote. Okay. So zygote having single cell undergoes mitosis, as all of you know. To certain extent, after having undergone mitosis, wherein cell division takes place, number of cells will keep on increasing, then the zygote will undergo different stages like morula, blastula, gastrula, like this. And at one particular stage, the zygote having converted into embryo will undergo what is called cell differentiation. That means basically, you know, zygote is a spherical structure, round structure. So when the round structure undergoes mitosis, naturally you expect all round cells. Correct? Imagine if all the cells would be round and zygote becomes embryo, embryo becomes an organism, what would be the shape of the organism? All would be footballs and basketballs. But we are seeing different shapes. Look at our shape, look at tiger shape, look at the plant's shape. So varieties of shapes are there. How is that is attained? It's attained thanks to cell differentiation. After the blastula stage, after the morula stage, in fact, a stage comes where the round cells will start undergoing differentiation. What do you mean by differentiation? Change of shape and size. So some cells become rectangular, some cells become triangular, some cells become cylindrical, some cells remain spherical, as Naveen said. And once a spherical cell is there that undergoes mitosis, bunch of spherical cells will come. One rectangular cell undergoes mitosis, bunch of rectangular cells, like that. So cell differentiation, thanks to it, it makes varieties of tissues, and that is why we said origin. If there is bunch of rectangular cells are to be formed, basically there should be a rectangular cell. Okay, that is the meaning of that origin. And why should embryo having spherical cells should undergo cell differentiation? What happens if all the cells are round? One, just now I said, we will be losing our shape. All the organisms would have been footballs and basketballs. But that is not happening, thanks to God for His creation. Different shapes are there, varieties of shapes and sizes are there. We are seeing every day. So thanks to cell differentiation. And when I said that spherical cell from the embryo morula stage undergoes cell differentiation and forms varieties of shaped cells, what is the purpose of that? The purpose is to perform a specific function. Okay? Imagine the function that a spherical cell will be doing in our eye. Compare that with a rectangular cell present in our thigh, or a cylindrical cell that is present in our muscle. Are they not different? Reverse you think. There is a rectangular cell in our eye instead of round cell. What would be the shape of our eye? Rectangular shape. And all of you are studying physics. You say F, focal length. What would have happened to focal length if all would have been rectangular cells in our eye? Will we be able to see how we are seeing today? Things would have been different. And some of the English movies you must have seen, isn't it?
Sir, boats have rectangular eyelids.
Eyelids, but eyeball? We are focusing on the eyeball, isn't it? Structure of eye you have seen in junior classes. And in physics you have talked about focal length, F. And I am sure you have moved the object either front or back, and image that is formed, isn't it, in 9th class physics? So as the object moves, the focal length also changes, and that's why when the things are far away we are having blurred vision, but as we approach we are getting clarity of the object. And some of you are wearing specs. Why are you wearing specs? Because of either myopia or hypermetropia, short sight or long sight. What for you are wearing specs? To correct that myopia and hypermetropia, isn't it? So basically,
Yes, sir.
If a cell is having a particular shape, particular size, it is designed to perform a particular function. There is specificity. It's not that just cell is becoming rectangular, cell is becoming polygonal, cell is becoming cylindrical. A round cell is attaining varieties of shapes to perform a particular function. That's why we define tissue as group of similar cells. When we say similar, size is same, shape is same. Similar cells having specific function and origin. Because of their origin, they get the size and shape. Because of their size and shape, they perform a particular function, specific function. Just reverse also you think, if all the cells that are present in our thigh, if they were to be round, will the thigh bear all the weight of our body? You know, our two legs are bearing the entire weight of our body, particularly knees, isn't it? So imagine if they were not to be strong, only spherical cells, what is our fate? So thanks to the cell differentiation. So based on size, based on origin, based on duty that it is doing, function it is performing, animal tissues are of 4 types. Okay. First one is epithelial tissue. Epithelial, as the name suggests, epi means upon, thelium means layer, it's a Latin word. So layer that is present on top of our body, which is the layer? Obviously it is skin. So the tissue that is forming our skin or any animal skin is epithelial tissue. Parallel to this, I told you, if you recollect from your memory, in case of plants, it is epidermal tissue. In animals, we are using the word epithelial tissue. Okay. Second one is connective tissue. As the name suggests, it connects one part with another part. Like all of you know, particularly sports boys, those children who are playing sports and games, you know, particularly basketball players, football players, hockey players, you know it. Bone to bone, one bone is connected to another bone with ligament. So sometimes, unfortunately, ligamental rupture will be there, or sometimes the ligament itself gets cut. Then what happens? Dislocation takes place. Unfortunately, some of us have got that experience. So it takes a lot of time. You need to undergo operation to put an artificial ligament, and then only the ligaments will be strong. The leg will become strong. You are able to move. And then, of course, one classical example that almost every day we speak is our own blood. In blood, as all of us know, there are 4 parts: RBC, red blood cells; WBC, white blood cells; platelets, and plasma. All these 4 are connected with each other. So blood is the classical example of connective tissue. Okay. Similarly, you must have heard about the word tendon. It is the connecting link between muscle and bone. Okay. So tendons are very important. Ligaments are very, very important. And of course, our own blood. Without blood, we are nothing. And the third one is, I am sure all the boys' favorite, muscular tissue. You are seeing so many Hollywood and Bollywood movies. Hrithik Roshan. Who is that? Some more heroes are there, no? Who are very, very fittest people. Building a lot of muscles, triceps, biceps, all that. So, muscular tissue. And you know, muscles are very important for us. They give shape for us, and they give toughness for us, mental strength as well as physical strength. So muscular tissues. And then last one, most important out of all, neural tissues, nervous tissue. And thanks to this tissue, because it is forming all the nerves that are running across in our body, plus our own brain. You know, sometimes we teachers call you when we get angry, you brainless fellow. But all of you know, being biology boys, you got a brain. Only thing is you are not using sometimes. So that brain is made up of neurons, nerve cells, right? We are going to talk about neural tissues. Now let us go with the first one, epithelial tissue. As I said, epi means upon, top of. Epithelium means layer. So epithelial layer is the membrane that is present on the surface of the body. And look at the shape, look at the number of layers that are present. So it has a free surface that faces body fluid or outside environment. It is the outermost layer. It covers the whole body as well as body parts. And cells are compactly packed with little intercellular matrices or spaces. That means they are closely arranged. There is no gap. That's what we say, compactly packed. And again, based on the shape, location, we say there are two types of epithelial tissues. Simple epithelial tissue, compound epithelial tissue. So what is the basis for this? In simple epithelial tissue, it is only one layer, single layered structure. Whereas in compound, it is multi-layered. Okay, many layers of epithelial tissue will be there. Now, going ahead with simple epithelium, it is composed of one layer of cell only, as you are seeing, and it lines body cavities, ducts and tubes, right? That means we have got in our body, as all of you know, coelom. We said true coelomic animals, body cavity, and we have a lot of ducts, as all of you know, and of course many tubes are there, including in our digestive system. So again, based on the function that it performs, simple epithelium is classified into three types: squamous epithelium, cuboidal epithelium, and columnar epithelium. Take care of the spelling: squamous epithelium, cuboidal epithelium, and columnar epithelium. As the names suggest, it is easy for you to understand where they are present and what is their function, right? Now let's go first one, squamous epithelium. Now most important thing here you must remember is, where are they present? So it is a flat structure, cells are all flat, thin layer, because it is only one layer, thin layer of flattened cells with irregular boundaries. There is no regularity, irregular boundaries. Where are they present? Found in the walls of blood vessels and alveoli in the lungs. What is the function? Diffusion. So they help in diffusion. Having present in the blood vessels and alveoli, they help in diffusion. Look at that. Oxygen coming inside, carbon dioxide going outside. So this is how they are helping in the process of diffusion. And once the oxygen gets inside, gets into the red blood cells, that blood, which is until then impure blood, becomes pure blood. Okay, purified blood. Why it becomes purified? Technically speaking, in RBC there is a pigment, red colored pigment called hemoglobin. So hemoglobin combines with this oxygen, becomes oxyhemoglobin, and presence of oxyhemoglobin makes the blood pure. Similarly, when carbon dioxide is there in the blood, in the absence of oxygen, carbon dioxide enters into the RBC, and hemoglobin now has no choice but to accept CO2 in the absence of oxygen. You see, there is a preference. In chemistry, your chemistry teacher will be telling hemophilic, okay, hydrophilic, like that. Phili means love, affection towards a particular thing. Hemoglobin has got lot of affinity for oxygen. So given a chance, if oxygen is there, readily it combines with oxygen, becomes oxyhemoglobin and becomes pure blood. But unfortunately, in the absence of oxygen, it has no choice but to accept carbon dioxide. Then it becomes carboxyhemoglobin, which is the indication of impure blood. So we don't like to have impure blood in our body. Always we need to have oxyhemoglobin. So what to do? Take lot of oxygen in and try to put the carbon dioxide out. And this is how we are doing in what we call deep breathing technique, isn't it? And that is the reason why meditation and deep breathing in yoga is very important. There are a lot of science involved in that. So the moment carbon dioxide is out, which is responsible for bad blood, oxygen is in, pure blood will be formed. So remember, squamous epithelium is present in alveoli and blood vessels. Now we move ahead with the second one, cuboidal. As the name suggests, cube, shape, cuboidal. Now, where is this cuboidal cells present? Now let us see that first. As we expect, it is composed of cube shaped cells, as you are seeing in the diagram. It is found in the ducts of glands and tubular parts of nephrons. What is nephron? Any idea, Naveen?
It is cup-like shapes, sir, which filter the blood.
Okay, where is it present? Sachin, Pallavi.
Kidney, sir.
Kidney, very good. Nephron is present in kidney. So we say it is the excretory unit of a kidney. And don't get confused, Pallavi, with neuron. Neuron is a nerve cell. Nephron is a excretory cell. So nephron is different, neuron is different. Okay, clear? So nephrons are present, as Naveen said, they have a particular shape, one head region and very long tail region. Okay, maybe in excretion chapter we will talk about that in detail. Now at present you remember, nephron is the unit of excretion present in kidneys. And presence of nephron is an evolutionary feature. And what does it do? Secretion and absorption. It secretes certain minerals and not only secreting, it absorbs. Okay, where exactly is present in nephron? PCT. There is a tube called proximal convoluted tubule. It is abbreviated as PCT. So epithelium of PCT is made up of cuboidal cells, and they are also present as microvilli. Now let us see the diagram. This is what, Naveen? Nephron is. And let us identify where is this PCT. I am just expanding it. Look at that. I think you are able to see the PCT, proximal convoluted tubule. Okay, this portion is PCT. So you got a cup shaped structure, and immediately after that cup shaped, teacup shape, it's called Bowman's capsule. Okay, Bowman's capsule. Just below that you got a tube with twists and turns. That is the PCT, proximal convoluted tubule. And when we take the section of that PCT, look at that, microvilli. And similarly, you can see the transfer section of that. So Bowman's capsule, transfer section. Now you know why they are present. Since nephron has to do lot of filtering, that means all the waste materials are to be filtered out from the blood. That is what we mean when we say excretion, isn't it? All the waste materials, poisonous materials, toxic materials, they all have to be removed from the blood. Otherwise, blood cannot afford to keep those toxic materials for long period inside us. It will ultimately affect us. Hence, there has to be filtration. So, during this filtration, I will use one word, osmoregulation. The kidney has to maintain, in fact, the nephrons have to maintain osmolarity or osmoregulation. I think sometime back I told you I defined osmosis. So, when the osmoregulation is done, then only all the toxic substances from blood will go out of our body. And sometimes it so happens, unfortunately, some useful materials like glucose, glucose is the ultimate source of energy for us. Whatever we eat gets digested, absorbed and forms glucose. And that glucose combines with the oxygen that we are breathing in and forms ATP. ATP is the chemical form of energy. Expansion of ATP, Naveen? Adenosine triphosphate. Okay? So, thanks to this, we are getting energy, we are able to absorb and reabsorb, escaping glucose, escaping minerals and even water to certain extent. So, all this in details I will be telling you in excretion chapter. Now, we go to the third one. What is the third one? Epithelial tissue, and the third one, columnar. Okay? Remember, we are talking of simple epithelium only. Okay? Columnar. It is composed of tall and slender cells. Cells are very tall and slender, means thin. Their nuclei are located at the base. Look at the position of the nucleus. Where are they situated? They are at the base, bottom. And free surface may have microvilli, hair-like structures will be there. Just now I have shown you microvilli. And it's found in the stomach, intestine. So, thanks to this tissue, columnar epithelial tissue, because of this, we are able to do both secretion as well as absorption. Let us see the figure here. We are taking now the transverse section of the intestine and stomach. This is how it looks like. Look at that. There is a lumen, means cavity, intestinal tract, and look at the cells. This is how the columnar cells will be present. Okay. Next, we go to next tissue, that is ciliated epithelium, that is modification. Sometimes the columnar cells or cuboidal cells will undergo certain changes. Then we call it as ciliated epithelium. Cilia, remember, in paramecium, hair-like structures will be present outside the paramecium. I hope you have recollected. You have seen in our biology lab under the microscope. Paramecium. Did you see in your junior classes? Okay, ciliated epithelium. No, if you are not seen, when you come back, first thing that you have to do is see that. We have got permanent slides, just observe them. Okay, so we go to modifications. Modifications, slight changes in the tissue. In ciliated epithelium, as the name suggested, cilia will be present. Now I am just expanding that. You can see the presence of cilia. Look at that, hair-like structures. Small hair-like structures, look at that. These are the cilia. So, having cilia, what do you expect? The function. Cells bear cilia on their free surface, present in the inner surface. Now, I want you to focus on the word inner surface, not outside, of hollow organs like branchioles and fallopian tube. In reproductive system, female reproductive system, there will be a fallopian tube which runs across. So, in that, and what are branchioles? In respiratory system, you are seeing here. See, I am expanding that. Yes. So this is the bronchus. Okay. Trachea is there. Below trachea, trachea is dividing into two parts. One part going right, another part going left, entering into the lungs. And the trachea is dividing into smaller tubules. Those are what we call bronchioles. Bronchus and further division bronchioles. So their function to move particles or mucus in specific direction over epithelium. So it is just like you are blowing them into a particular thing. And having done that, next modification is columnar or cuboidal cells will also perform sometimes like glands. Then we say glandular epithelium. What is the function of gland? First of all, you should know what is a gland. Any idea? Can someone define a gland? Yes, Naveen, Pallavi. It produces hormones. Okay. What is it made up of? You are telling function. Good. What the gland is made up of? That's all cells. It's made up of cells. So group of cells that perform secretions. Isn't it? A gland is secretion mainly. So sometimes this epithelial tissue will start secreting something, having a particular shape. Then we say that epithelial tissue is modified and become glandular epithelial tissue. So basically this has become glandular epithelial tissue just to secrete something. Like all of us there are two types, unicellular and multicellular. Unicellular, only one. It consists of isolated glandular cells, only one cell. Like we have got a goblet cells of the alimentary canal. Then alimentary canal means digestive system. Okay. Then multicellular, it consists of many cells, group of cells. Like example, salivary gland. It secretes saliva. Like when Naveen is very hungry and his mother is preparing laddus, far from seeing the laddus only, our Naveen gets saliva. Who is doing that work of secreting saliva? It is the salivary glands that are present inside us. So under the tongue, saliva. Okay. Next, the glandular epithelial tissue. Based on mode of pouring their secretion, glands are of two types. Now we know that gland is a group of cells that secretes something. Some juices are secreted, be it hormone or anything. So where exactly they are secreting? Is it outside or inside? So based on that, glands are of two types. That is exocrine glands and endocrine glands. Look at that. Exocrine, E-X-O-C-R-I. Endo means inside. They secrete inside, endo, and they are present inside also. And exocrine means they have ducts, one tube will be there. So first basically one gland will be there. What is a gland? Group of tissue. And the tissue will have one tube. Okay. And that tube, if tube is there, then we say exocrine gland. On the other hand, only tissue is there, gland is there, but there is no tube, duct is not there. Then we say endocrine. So the other name for this endocrine is ductless. There is no duct, ductless glands. Okay. So which is advantage, exocrine or endocrine? Obviously endocrine, because whenever they secrete hormones, endocrine glands having no duct, 360 degrees they secrete the hormone. Whereas exocrine, even though it secretes hormone, it has to travel through the duct and reach the destination. By the time it reaches the destination, lot of time is taken, time consuming, isn't it? So always having endocrine is fast. So exocrine glands, they release secretions through ducts or tubes, and exocrine glands secrete mucus, saliva, ear wax, oil, milk, and all the digestive enzymes. Whereas endocrine, ductless gland, they produce hormones directly into the blood. That is the advantage. Okay, I think we are running short of time. Tomorrow's class I will cover compound epithelial tissue. If you got any doubts, post your doubts in our WhatsApp group. Until then, take care. Bye.
Thank you.